Welcome to CognitionChunks. This information is for entertainment only. I'm not trained in genetics. Is this useful? Is this nonsense? Join me as I attempt to learn from GPT. PhytoSynthetica. Can GPT design a new organism for positive climate change? Episode 14. Case studies of successful biomass to biopolymer conversions in engineered organisms. Welcome to our deep dive into the fascinating world of synthetic biology and sustainable materials science. Today we're exploring groundbreaking case studies where scientists have harnessed the power of engineered organisms to convert biomass into biodegradable polymers. These advancements promise a future where eco-friendly materials replace traditional plastics, significantly benefiting our environment. Case Study 1, Polyhydroxyalkanoates, PHAs. Production in Cupriavitis necator. Overview, Cupriavitis necator, formerly Ralstonia eutropha, stands out as a model organism for PHA production. Naturally, it accumulates PHAs as carbon and energy storage compounds, particularly when nutrients are limited, but carbon is abundant. Engineering approach. Scientists have turbocharged C. necator's natural abilities. By overexpressing genes like PHA synthase and β-ketothiolase, they've ramped up the organism's capacity to synthesize PHAs. Additionally, they've introduced new substrates to broaden its production base. Results? These engineered strains now achieve PHA contents exceeding 80% of their cell dry weight, showing remarkable yield and productivity. Even more impressively, they thrive on diverse renewable substrates, including waste plant oils and lignocellulosic hydrolysates. This versatility underscores C. necator's potential as a sustainable biopolymer factory. Case Study 2, Polylactic acid or PLA precursors in Escherichia coli. So, PLA is a biodegradable polymer that finds use in everything from packaging to medical devices. It's typically made by polymerizing lactic acid, which can be fermented from biomass-derived sugars. Now here's the interesting part. Researchers have reprogrammed E. coli to produce lactic acid directly. By overexpressing lactate dehydrogenase and tweaking metabolic pathways, they've maximized precursor availability. The results? Engineered E. coli strains now churn out high levels of lactic acid from glucose and other sugars. Some strains are even fine-tuned to produce specific forms of lactic acid like L or D. These forms are crucial for tailoring PLA's properties. These advances pave the way for more sustainable and efficient PLA production. Case Study 3-poly-3-hydroxybutyrate or PHB in Synechocystis sp. Cyanobacteria like Synechocystis sp offer a unique advantage. They can convert carbon dioxide in sunlight directly into valuable compounds, including PHB, which is a type of PHA. Genetic modifications have focused on diverting carbon from photosynthesis into PHB synthesis. This involved introducing PHB synthesis genes from other organisms and blocking competing pathways to enhance yield. The engineered Synechocystis strains can now produce PHB from carbon dioxide and light, acting as solar-powered bioreactors. This represents a major leap toward technologies that capture and convert atmospheric carbon into useful biopolymers. Case Study 4. Hybrid biopolymers in Saccharomyces cerevisiae. The yeast Saccharomyces cerevisiae, well known for its role in baking and brewing, is also a promising candidate for producing hybrid biopolymers with customizable properties. Researchers have constructed multi-gene pathways from various organisms within Saccharomyces cerevisiae. These pathways synthesize novel monomers, which are then polymerized into hybrid biopolymers. This sophisticated engineering involves dynamic regulation and compartmentalization to optimize production. These yeast strains have successfully produced novel biopolymers with diverse, tunable properties. This opens exciting new possibilities for biomanufacturing materials suited to specific applications, from biodegradable packaging to medical implants. These case studies illustrate the incredible potential of engineered organisms in transforming biomass into biodegradable polymers. Through sophisticated genetic and metabolic engineering, We can harness the metabolic flexibility of microorganisms to produce a wide array of biopolymers, offering sustainable alternatives to petrochemical-based plastics. As research and development continue, we can expect an expanding diversity and applicability of biologically produced polymers, significantly contributing to environmental sustainability and green technology advancement. Don't forget to like, subscribe, and hit the bell icon to stay updated on our latest content. Thank you for joining us for another Cognition Chunks.